Hello, my name is Linlin Zhao, and I'm assistant professor at the Department of Chemistry and Biochemistry of Central Michigan University. I'd like to spend a few minutes to explain to you about our research and how basic biomedical research can have a positive impact on our life. Cancer is the second leading cause of death in the United States. Cancer costs billions of dollars in the nation's healthcare bill. Cancer also costs lives of the people we love. An important aspect of cancer formation is exposure to substance that can cause cancer, also known as carcinogens. Take smoking, for example. Tobacco smoke is known to contain more than 70 different carcinogens, and smoking is known to cause a number of human cancers. A second example is exposure to aflatoxin, common food contaminants in peanuts and corns produced by fungi. Exposure to aflatoxin is known to associate with increased risk of human liver cancer. The overall goal of my research is to advance the understanding of the molecular mechanism of cancer formation in order to improve the efficacy of cancer treatment. So how does carcinogen cause cancer? Cancer formation is a very complicated biological process. Fortunately, research over the past several decades has made remarkable progress in understanding the different causes of cancer. Take carcinogens, for example. It is now known that many carcinogens are able to damage the genetic composition of our cells, also known as DNA. The damaged DNA can lead to wrong genetic instructions of cellular metabolism. In other words, carcinogen-induced cancer formation often involve changes to the genetic composition of our cells, which can throw the regular metabolism off balance. Therefore, carcinogen-induced DNA damage is an important driving force for the initiation of cancer formation. Fortunately, there are many protective mechanisms to help fight against DNA damage in our body. One aspect of research is to study a protective mechanism known as DNA damage bypass. This is a biological process that helps to accomplish DNA copying process despite the presence of DNA damage. Why DNA copying process is important? Because the same set of genetic material has to be duplicated for every new cell formed in our body. During the DNA damage bypass, specialized DNA copying enzymes are involved to rescue and to overcome DNA damage, which could otherwise block the DNA copying process. If we compare the regular DNA copying process to taking a road trip, DNA damage is like a sharp metal piece on the road, which could punch our tire and stop our vehicle. DNA damage bypass machineries or enzymes are like emergency service vehicles that help to rescue and to fix the problem and to help us get back on track. However, there are two sides to every coin. If you remember the title of this presentation, the DNA damage bypass process is a double-edged sword. This is because the rescuing process is not always accurate. The DNA damage bypass process itself can sometimes introduce wrong genetic codes, which in turn contributes to cancer formation. A second, but equally important aspect of the potential negative effect of DNA damage bypass is related to cancer treatment. Many cancer chemotherapies kill cancer cells by damaging their DNA. And the bypass machineries existing in cancer cells can sometimes protect them from DNA damage, which leads to chemotherapy resistance. Both of these aspects emphasize the importance of understanding of these machineries and their relevance to human cancer. My laboratory is investigating the mechanism of action, structure, and competency of several of these molecular machines. By gaining fundamental knowledge of these biomolecules, we hope to bring our understanding of cancer formation to a higher level. With the new knowledge learned from our research, we hope to inform the development of more effective cancer treatment and to help fight the battle against cancer. Thank you for listening.